presentations today. Actually, there were a couple of very nice questions about the importance of embryogenesis, of gastrulation during embryogenesis. So you have to look at this from the lens of genetics as well, because genetics and regulation is going to play a key role into defining how is the embryo, how is the vertebrates of the embryogenesis of vertebrates. Linking with my presentation before, we have 5 embryos here. Wild type. This is wild type embryos that develop normally, and then you can see how it's more or less the development of an embryo in 24 hours.
Mr. David.
Yeah.
Sorry to interrupt you. Can you just make it a presentation mode? Full screen?
It's full screen now, yeah. I don't know.
Okay.
It's full presentation.
Okay, yeah. Please go ahead. Yeah, okay, please go ahead.
So if you want to compare this with another type of embryo, we can look at a mutant that does not develop the embryogenesis, the gastrulation as well. This is a mutant. You can see that the development is a little bit different. And this is a single pin-eye embryo. It just developed one eye. It's a mutant, indeed, of several feet. And you can see that it doesn't develop 2 eyes, but 1 eye only. And this is because the regulation and the gene expression is different. We can see this again. It's very interesting and important to see the difference. You can see that at the beginning it develops more or less similar, but then at this point of gastrulation, the gastrulation is different, and because of the gastrulation is different, the embryo doesn't develop 2 eyes. If you want to see the same movie again for wild type, you can see here 5 wild type embryos compared to the mutant. And then you can see that the gastrulation is more or less similar in all of them, and they develop 2 eyes, all of them. But the orientation is a bit different. It's a difference of the embryos. So we define a workflow to do this, following with the previous presentation. We have at the top row, we have the raw data. Then we develop the digital representation, which is the displacements of the cells between 2 different images, and then the trajectories. And from the trajectories, we develop the Eulerian indicators, that is the topology of the deformation. And then we also see the patterns in time, how they develop the cumulative displacement. And the tool that we introduce here is Omix Representation, because then we can express in this figure that there is more like a heat map. Each line corresponds to a trajectory, so we can see this is a profile. This Lagrangian representation is profiles, and from these profiles we can see through time which is the compression, expansion, and deformation of single cells along time. And then we can see that they compress at the beginning and they expand a little bit later. But if we see the cumulative behavior, it's compression, because the expansion is always smaller than the compression. So it starts compressing and keeps compressed the cells along time. So there is during gastrulation, there is a very strong process of compression to make the gastrulation, and this compression process is different, as we will see, in mutants and wild type embryos, and this is because of genetic expression. And then the last tool that we introduce at the bottom row is the categorization. It's the categorization because we can make similar profiles, and every single profile will be a different Lagrangian biomechanical profile, canonical. So we can make a categorization and reduce the dimensionality of all the mechanics information and all of the genetic expression to have profiles we will see later on. So as we were saying before, we have the deformations here. Very interesting. You have the velocity field and the compression and expansion. The compression is red and blue, and the distortion is in blue and golden, and then the rotation is in blue and green here. We can see this again. This is the normal behavior of zebrafish embryo. You can see it again. This is how it normally develops the gastrulation and the vertebrate and the embryogenesis of vertebrates. And then we can see here the maps again. But if we make a comparison, we can see that the patterns between the wild type in the top row and the mutant in the bottom row are different. It's much less organized, but it's also delayed. The embryogenesis that you see at 8 hours and at 8 hours between the wild type at the top and the mutant at the bottom is very different. The embryo is developing slower, and because it's developing slower, the compression pattern in blue and red is different in the top and in the bottom. So this is going to change the gastrulation, and you can see here more. You can see here better in the patterns. What you see, and you see the compression is generating a border, and it's not creating a bilateral symmetry between the two halves of the embryo. You can see here again in green, in blue and red. You see that the compression is delayed, and when it's delayed, instead of having a symmetry between two parts, it's more like a barrier between the bottom and the top. And that because of that compression, the embryo doesn't develop the same way. So the embryo scale patterns in compression is changing completely because of the genetic expression. And we can see here a comparison time to time. And then we can see here, as you see at 11 hours, that there is a strong compression that we've seen before in the movie. And it's going to change the whole embryogenesis. We can see the same here in the Lagrangian pattern. We would see the normal behavior of the embryo in the Lagrangian patterns. Through time evolution, there is a strong compression, as you can see in the second row in the J indicator. But if you see the same pattern in the mutants, you see that there is a strong difference. And this is because in the wild type, we have this information here that you can see that is the guscoid expression, guscoid gene in green. This gene accelerates the embryogenesis and the dynamics between the epiblast and the hypoblast. You can see the movie again, maybe. Because of the genetic expression, the compression is very well conducted through the embryogenesis, and the compression is properly done, and there's bilateral symmetry that is generating the eyes. And the guscoid expression is leading the creation of the eyes of the embryo. You can see here the dynamics between the hypoblast and the epiblast. You see two different layers. Below, the hypoblast is going faster, and this is generating a different type of compression because in the mutant, when there is high compression, the hypoblast doesn't move that well. And then you can see, and with tools that we develop, we can see this because we can label the hypoblast in red and the epiblast in blue. You see the two tissues? And then you can see that they have different dynamics. The compression and the expansion is different. And how we can measure it? By profiling the two tissues, making this a profiling of the two tissues. And then we can see for different embryos, every column is an embryo, and then every column is a descriptor. And then you can see that they have some commonalities for all wild type embryos, that the patterns are kind of similar, even when they have a strong variability. So this is very important because even with the same phenotype, a wild type phenotype, the patterns are even different. But if you compare with a mutant, it's completely different from 8 hours when we will see before, that the compression is so different that it changes the whole development of the gastrulation, because there is not, in the mutant, there is not guscoid expression. So the difference in the embryos is that for the mutant, there is no green information here. There is no expression of the gene. So it's very important that if we remove the gene expression of the gastrulation of the guscoid and other genes, the development of the embryo changes completely. And you can measure it by profiling the different trajectories of the cells, as we are seeing here. So we can label two tissues, and we see the tissues over time, and we can have the profiling of these tissues in terms of mechanics. Even more, we can categorize this profiling by extracting different canonical profiles. This is the lines that you can see here. You have the whole profile, but then applying machine learning, we can identify 3 characteristic profiles for each kind of descriptor. And with this, we can go even further and make an Omix representation in which every cell is labeled with a characteristic behavior, depending if it belongs to a canonical profile. It's an omics characterization that we can make mechanomics. Mechanomics will be the term. We can make mechanomics and compare it with the genetic expression. If you compare the mechanomics of the wild type of the mutant, you can see different populations. You see that green population. That green population is related to the guscoid genetic expression. That is different mechanics. But if you compare that in the wild, in the mutant, that green population doesn't exist because there is no genetic expression at this point. Therefore, the morphogenesis is completely different, and the difference is that the wild type develops normally, and the mutant only develops one eye. If you see this labeling of mechanomics, you can see here that the green population follows a pattern that is more or less similar to the guscoid expression, and it's organizing, and you can see more or less the eyes and different population within the eyes: red, blue, no, red, yellow, and purple. But if you see this same movie for the mutant, you can see that there is no green population, and the red population is mainly the eye now. It's much more compression, and it doesn't develop properly. So this is different dynamics that are characterized because of the genetic expression. We can see the genetic expression one more time. As you will see, the green expression here, which is from the microscope, is just a marker of genetic expression of a gene. It corresponds more or less to a mechanomics, a specific population of the mechanomics. So there is a correspondence between genetics and mechanics. And you can see here in this population, because we have made the same profile of genetics in the number D, we have 3 different populations that you can see at the beginning. And there is one that is a lot of genetic expression at the beginning, and then it keeps growing. And then you have other 2 populations, one that one population that has a little bit of genetic expression in the middle, and one population that doesn't have any sort of genetic expression. If you compare the D and E, you can see that there is some commonalities between D, which is the profiling of the genetic expression, and E, which is the classification of mechanomics. And this is more or less the message that we want to convey here, that in embryo patterns, every genetic expression can have a relation with the mechanics in terms of mechanomics population. Thank you. Thank you to United Research Forum for this space for the presentations.